Yeah, so this morning I'll be telling y'all about some efforts I've been working on for my PhD research, where we are developing models to be able to predict the engraftment of C. diff. So why do we care about Clostridioides difficile or C. diff? Well, C. diff is a leading cause of GI tract infections. While this is a global problem, it's particularly a significant issue in the US and countries like Australia and parts of Europe. In the US alone, around 500,000 people are affected, and around 6% of these infections can be fatal um, due to severe diarrhea and other complications. And the risk factors for this disease or this infection are typically the use of antibiotics in the clinical setting, uh, or any sort of perturbation that can lead to the wiping out of your native microbiota. And there's been a lot of progress actually in developing ways to combat this, but so far the most effective has been the fecal microbiome transplant, wherein an individual receives a fecal sample from a healthy donor that can allow them to sort of recolonize their native microbiota. In this context, the use of antibiotics has been shown to be effective in some cases, but generally not an ideal remedy. And a lot of research has been done on this topic, and so there have been numerous studies that have found various associations with the abundance of certain microbiota with the prevention of disease. And so, but there's still a lot of work to be done to fully understand this and develop probiotics. And so, but the C. diff, the story of C. diff is an interesting one because while it does present a risk to the host if CDI or C. difficile infection develops, it can also colonize many of us, and it does colonize many of us, and can simply be there and be fairly benign. And when you have a healthy microbiome, some estimates suggest that maybe 4 to 15% of us can be colonized by C. diff and be asymptomatic carriers. In infants, actually, the rate of colonization is much higher. Many infants have C. diff in their guts, and then as they develop, they lose C. diff. And really, the trajectory from colonization to infection occurs when there are perturbations, as I mentioned. And part of the reason this is because C. diff is a spore-forming organism, and those spores can persist following disruptions, allowing it to survive the disruption and then bloom in the absence of any competition. And so to try to address this issue and kind of add to the body of research that has been done, myself and members of the lab developed a modeling technique. And this is the sort of workflow that encompasses the research I'm describing to you this morning. We essentially took publicly available data and developed an in silico invasion assay to be able to, in combination with a metabolic modeling technique, to be able to answer the question of is C. diff predicted to invade certain microbial communities more often than others, and then use the predicted, in addition to the predicted growth rates, leverage the predicted metabolic profiles to then try to understand the strategies and niches occupied by C. diff when it does successfully invade. So as a first sort of test of this technique that we've developed, we actually we leveraged some data where an individual had actually been shown to be colonized by C. diff. Here I'm showing you a time series from one individual where there were samples collected nearly every day for about, you can see 200 days. And what had happened actually is you can see in this red portion, this person actually got food poisoning and had a diarrheal event. And you can see in the plot below that they did not have any C. diff. And then following this diarrheal event, they became colonized with C. diff, as indicated by the 16S relative abundance in the blue trace. Now, when we take the same data and we apply our modeling technique, it's called MICOM, what we see is that actually the predicted growth rate for C. diff in these samples mirrors that of the relative abundance. While you see it's not quantitatively like exactly overlapping, you do see a very similar trend in that prior to this diarrheal event, it's not predicted to be able to invade. And then following that event, you see the individual become susceptible. Furthermore, there's this data set included another individual who similarly had an event where they got food poisoning. In this particular case, you see from the 16S profile that this individual was already colonized with C. diff, albeit at a very low level, hence the sort of spikiness of the trace. This individual had C. diff kind of close to the limit of detection. And then similarly, while you see quantitatively these results are not very well correlated, you see a similar qualitative trend in that the model is predicting that throughout time, this individual is or could be susceptible to colonization. This kind of gave us an initial validation that this technique could work. And so we went forward and leveraged publicly available fecal microbiome transplant data. These are data where individuals who had experienced the CDI came into the clinic and then received a FMT to then try to alleviate their infection. When we apply our modeling technique to these data, what we see are that individuals prior to receiving an FMT are nearly all predicted to be able to be invaded by the pathogen. Following FMT and then in the transition to the healthy state, we see that while still a decent proportion of individuals are still predicted to be susceptible at different points in time, you do see that the distributions become bimodal, and there are a significant fraction that have very low predicted growth rates to the point where they're essentially not growing. When we compare these predicted growth rates with the empirical diversity of the microbiome, we see, as expected, that the individuals coming into the clinic pre-FMT had very low Shannon diversities, and that following FMT, you see a recovery in the diversity that is indicative of being colonized by a recovery in their microbiome that can lead to a transition to a healthy state. What's interesting about this modeling technique is that in addition to predicted growth rates, we're able to obtain predicted metabolic fluxes for all the organisms in the community. And so here I'm showing you 4 individuals' time series that are the individuals receiving FMT. What we did here and what I'm trying to show is that if you predict the consumption patterns of the microbiome in the absence of C. diff, what you find is that reducing that high multidimensional space into 2 dimensions using these UMAPs, the predicted consumption pattern of the microbiome, which is displayed on this UMAP, is actually predictive of the C. diff growth rate, which is the color axis for each point. And what you see is these clusters form where you see there's this sort of low growth rate cluster in the middle, and then 2 high growth rate clusters. that are suggesting the microbiome state is really important for the ability for C. diff to invade these communities. And now these kind of complicated looking trajectories are actually the patient trajectories, where you see they start with the red circle, and then they eventually end up at the red star. And so what you can see here is these patients, most of them start in a state where the growth rate, in a susceptible state where the predicted growth rate of C. diff is high, and then they sort of bounce around and generally end in a state where the predicted growth rate is low. To try to better understand the mechanisms that might be underlying the susceptibility of these individuals at different points in time, we further investigated the apparent metabolic strategies of C. diff in silico. To do that, we examined C. diff's specific import fluxes that it was using. And here in this heat map, I'm showing you the C. diff import fluxes across all the samples. Each row is a metabolite flux. Each column is a sample. And what we see is this color bar and the clustering indicate 3 different regimes, 2 of which are associated with high predicted growth rate. You see the first one on the left, and then a sort of more moderate growth rate on the right, and then a sort of low growth rate regime in the middle. And looking at the heat map, you can see these are associated with different import fluxes. You can see, for instance, on the left, there's a state where it achieves the highest growth and it's importing pretty much everything that it can. Whereas there's a more moderate growth rate state where it's importing many metabolites, but only not the full set. And this is indicating that maybe there are different niches that are occupied by C. diff in these samples. Furthermore, we see actually several metabolites that have been empirically associated with its growth in vivo that show up in these predictions. Those include trehalose and ornithine. Importantly, we also see that a number of sugars and amino acids seem to be making up the different set of metabolites that seem to be preferred by C. diff in these predictions. But then to further kind of like test or see whether or not these predictions were specific to this data set or whether or not they might generalize, we applied the same modeling technique to 2 additional data sets. David et al. Is the initial time series I showed you. Burkell et al. Is the one I've been describing now. Then we leveraged a large cohort, a cross-sectional cohort from individuals here in Seattle, as well as a large cohort that is now actually called the Microbiota Initiative. It was originally called the American Gut, that is based from a group in San Diego. And what we see here, again, this is a dimension reduction of the import fluxes. Now this is just for C. diff, not the community. And what we see across these 4 data sets is a very similar sort of profile that is formed. Importantly, it does sort of show that there's like a high growth rate regime, a low growth rate regime, and a sort of a midpoint, suggesting again that there's a spectrum of metabolic strategies and associated growth rates that can be achieved by C. diff across individuals. Leveraging this data, we could also sort of look at associations with the predicted growth rate and the diversity of the samples. And what's interesting here in this figure is you do see the sort of known trend in that you see that low diversity is associated with high predicted growth rate. And then as you sort of approach this like average, more moderate diversity, you see the growth rate, predicted growth rate decreases. Now, one interesting and sort of unexpected feature here, though, is this also suggests that at very high diversities, you actually get C. diff could achieve higher growth rates comparable to that of the growth rates at low diversity. And we know that very high diverse communities can be associated with states like constipation and other sort of non-ideal clinical phenotypes, suggesting that higher diversity isn't always better. And then finally, we to sort of further validate this approach and try to move towards our ability to maybe develop probiotics, we leveraged data from a study that successfully demonstrated a clinically relevant probiotic that is composed of about 8 organisms. Here I'm showing you some figures from that paper. The individuals here were able to show that this probiotic could colonize the human gut, and in mouse models was effective at increasing the survival of mice infected by C. diff. Here you see, for instance, that this in the absence of FMT or any sort of intervention, many mice would die, but then using this probiotic is actually more effective in this case than FMT. And so we leveraged the information here, the composition of this probiotic, to try to design our own simulated intervention using the data set that I previously described for the FMT recipients. And so essentially we do the simulated intervention similar to the simulated invasion. We just introduce the probiotic alongside C. diff in this case. We also introduced a simulated antibiotic intervention where we reduce the abundance of organisms known to be affected by vancomycin. And what we see is, again, across these growth clusters that I described previously, in the absence of any intervention, you see there's this sort of high, medium, and low growth regimes of C. diff. When you introduce the probiotic alone, you see a reduction in the average growth rate across the high and medium growth clusters, but there's a very high variance. The use of the antibiotic itself is fairly effective. Reducing the abundance of C. diff has a strong impact on its predicted growth rate. And then when you combine the antibiotic and probiotic treatments, you see the most effective outcomes, where in many cases, but not all, you can reduce the growth rate of C. diff to essentially a regime where it cannot invade. And this is in line with the results of this study that found that for the probiotic to be the most effective, individuals needed to take an antibiotic first to clear their microbiomes before it could effectively colonize. Examining these results a little bit closer, we see that essentially when the probiotic is growing, it is suppressing the growth of C. diff. When the average growth rate of the probiotic strains is low, you see C. diff is still able to invade. And when we look at the sort of average distance in their consumption profiles, we find that many of the probiotic strains, for instance, this Bladia producta.
Alex, still 1 minute.
Okay, I'm nearly done. You see that many of these probiotic strains share similar niches to C. diff. Their niche space is very close. And if you look at the niche space as a function of growth rate, when the niche is overlapping, the growth rate is suppressed. When it's not, it's high. And finally, when we look at the consumption profiles of these probiotic strains, you see many of them are overlapping with C. diff, which is on the left. And that when they essentially are competing with C. diff, which is the mechanism for suppression. And so in conclusion, I described this modeling framework for pathogen colonization simulation, and we were able to characterize some mechanisms of colonization by C. diff. And I think moving forward, these techniques can be used for a general framework for predicting engraftment and designing of probiotics. So I'd like to thank my advisors, Sean Gibbons and Nitin Baliga, as well as Christian Diener, a postdoc in the lab that developed the metabolic modeling technique, MyCom. And yeah, happy to take any questions.